Sí, sí, no, lo veo bien.
Es importante la decisión
Muy bien. Gracias. Hasta luego.
Vale. Fins ara. Hello, everyone. Sorry for the inconvenience. I'm Julia Crespin. Some of you know me. I'm an oceanographer and a climate scientist. I recently defended my PhD in physical oceanography last September. And right now I'm a postdoc in the Earth and Ocean Dynamics Department of University of Barcelona. I don't know if online they can hear me well.
Checking now.
Okay.
I'm asking them.
Okay, but I will present, okay, perfect. I will present basically the two models I developed during my PhD thesis. And yeah, that's all. I will do a quick introduction just in case some of you are not from the marine part. But yeah, here we have a NASA video of the atmospheric carbon being removed by plants, like the seasonal respiration of land. We always hear that the Amazon is the lung of the planet, but we forget that there's another part that is also very important, and this is the ocean. Here we see again another video from NASA, and we see ocean chlorophyll in this color scale that it serves as a proxy of primary production that occurs on the ocean. But what is marine primary production? It's basically the process by which phytoplankton converts CO2 into oxygen, which is the same as inorganic carbon, into organic carbon. But why is marine primary production so important? First of all, it represents the 50% of global primary production, so it's equally important as what happens on land. It also represents the base of marine food web, and in relation with climate change, it contributes to the carbon sink by the biological carbon pump. So CO2 from the atmospheres and end up stored in the deeper ocean. So which methods do we have for measuring or estimating marine primary production? The simplest, not simplest, but the most direct one is in situ data, ground-based data, which, as you can see here in this figure from Mattei and Scardi 2021, this data set gathers all observation of marine prime production observations that are public. And as like you can see here, these are sparse both in space and time. And then there's a lot of techniques, lab techniques to obtain these observations. And these are also very different. I won't go into detail into that because I'm not a lab technician, but they are diverse and these introduce a lot of uncertainty too. Then we have remote sensing algorithms, which give us a global coverage, but we have different models and each of them have a different assumption. The easiest, simplest one is the vertically generalized production model. And it's what we call a chlorophyll-based model because the main component is chlorophyll concentration from satellite observations. Then we have the carbon-based production models, which are mainly centered on carbon concentration that we derive from backscatter information from satellite images. And the most complex and recent one is the absorption-based models, which use absorption information from satellite. And this equation is from the CAFE model, which is the carbon carbon absorption fluorescence euphotic resolving model, which is a very long name. But this is the most recent one. It's from Silsbe et al. From 2016. And as you can see here in this figure from Brian Keogh et al. From 2023, depending on the assumption, if we use a chlorophyll-based, a carbon-based, or an absorption-based model, outputs in NPP diverge. Then we have biogeochemical models, which are more complex, and we have a lot of components, as you are probably more familiar with. This can give us also global coverage, but depending on the model, we have different assumptions and different components, and this gives us uncertainty in our parameters, and also it introduces us another complexity, which is the computational cost. Here in this scheme from Fennell et al. 2022, we can see that there's different complexity introduced into models depending on which components we add, phytoplankton, different nutrients, and so on. So we can introduce more complexity, and this can also give us more uncertainty. As you can see here in this figure from Ryan and Keogh, these are CIMMYT6 models, and we see that depending on the biogeochemical model, we get very different changes in NPP, in global NPP in the ocean. And finally, the most recent techniques, the most novel ones, are the ones that use machine learning or data-driven methods. This can give us global coverage. It can also capture complex interactions that are embedded in data that, as scientists, is very difficult to capture or to detect. But we depend on the quality and quantity of data that we have available. This also makes this type of methods very hard to validate. So here we have 4 different typologies of techniques to obtain marine primer production. And as we can see, there are some pros and cons in each method, but none of them is perfect. So the main objective of my thesis, my main goal, was to assess and enhance the quantification of primer production in the ocean, but also taking into account the context of climate change. So I wanted to assess the current limitations that we had in this quantification. That this is a summary of what I did, and then develop and implement new approaches, see the implications, the ecosystem implications that these new models give us, and finally assess the complexity of climate change into this quantification and take into account that we have to do efficient methods in this context of global warming. So the first model that I developed is the offline Fennell model, which is a biogeochemical model. I did most of the work at Dalhousie University with Katja Fennell, so it's her model originally from ROMS, and I will explain a bit. So this model evolved from the Coast ROMS oil model, which was developed from Fink et al. In 2021. This was a passive tracer offline model. So it was used for passive dyes, for oil spills, and it worked very well with ROMS. So we were like, okay, let's try this, but with the biogeochemical component. Why? We have the physics, and then we can run the biogeochemistry in an uncoupled way, so in a sequential way. And why offline? First of all, biogeochemical processes use a larger time step, so it's less computing time. Then most of the instabilities that come in simulation come from the hydrodynamic component. So if we first run the hydrodynamics with a finer time step, and then once we have this solved, we run the biogeochemistry, we will hopefully not have not have model crashes or simulation crashes, or not as much as we would have. Then this also helps us to reuse simulations. Once we have a hydrodynamic simulation, like the physics with circulation, temperature, salinity, we can do as many biogeochemical simulations that we want. And these allow us to do, for example, more sensitivity experiments or parameter testing only with one hydrodynamic simulation. So this, in the end, translates into less CPU, less computational time. And why did I use ROMS and offline FENL? I say the best of both worlds. So ROMS is a very robust and validated model at regional scales, also in climate experiments. And then also the FENL model is very recognized and widely used in many, many regions of the world. So we have the physical reliability, we also have this biogeochemical flexibility, and then we can include this computational efficiency of the offline. This would allow us to test different scenarios at optimized computational cost in the context of climate change. So this is the scheme of the offline model. It's at the core, it's the nitrogen, carbon, plankton, and detritus from Fenel et al. From 2006 and 2008. And then when I was at Dalhousie, I extended the offline model to include phosphate, oxygen, and also river detritus. This is what it's done in the model, but I don't have time to explain all of this, but it's basically, it does the same as the Fenel model that works in the ROMS model, that if you have used ROMS, so it's basically the same. We had to change the temperature and salinity reading because it had some problems. And then also free surface, so zeta. And as I said, I included the phosphate computations, the river detritus. And then when I was there, they had included the Wannikov computations of oxygen into the Fenel model. So I was, okay, I'm editing the code. Why not including these two in case someone also wants to do this? For example, river detritus, I usually don't use it, but I had the time, so I included it just in case the community wanted this. So you can activate and deactivate these options. And then for validating this offline Fenel model, I did this strategy. First, I did the model evaluation in the northern Gulf of Mexico, because the Fenel model has been implemented there in a lot of studies and tested a lot, because they have these hypoxia events, which are very frequent. So I wanted to test it there. And then I did a model ground truthing in the Mediterranean Sea. So the first study is this model evaluation, which was conducted in the Gulf of Mexico. This was published in September in Geoscientific Model Development. And what we wanted to see is how, what were the difference between a coupled, so an online simulation using ROMS and Fenel model, and our configuration of using the physical component of ROMS, and then in a separate way, the offline Fenel model. So we tested different time steps so we could see our computational gain. And then we also tested 3 vertical mixing schemes, which is the generic length scale, the large McWilliam-Donny, and the Miller-Yamada 2.5, just in case one of them was better than the other, so we could check. So in terms of computational time, here you can see in this line plot the different mixing schemes, and in the x-axis we can see the online simulations, so the 2 components, and then the offline simulations using the same time step, so por uno, I don't know, like times 1, and then times 3, so the 3 times faster, 5 times faster, and 10 times faster. So we had gain time, so it was less 80% time, computational time, so it was very good. And we were, okay, what's the skill score? How good is offline Fenel? So we computed the skill score, and as you can see here with the three vertical mixing schemes and the different time step multiples, all of them are between 92 and 93% skill score, which is very good. You can test different things, and then if you say, okay, 92% skill score is not enough for me, then I can do the simulation I choose, the simulation I want. But you can see different things with this skill score. So here you can see the time series. This is with the ×5 time step. And here you have with blue tones, the large McWilliam-Donny with, well, the different vertical mixing schemes. And I chose oxygen, phosphate, and nitrate. But as you can see, there's basically no difference between the offline and the online simulations. Here, as an example of the spatial concentration of chlorophyll, if we see the difference, there's some bias. So it's not perfect. But then if we look at the online simulation and the offline simulation, if we look at this green color scale, I would say it's not much of a big deal. So then we implemented the model in the Mediterranean Sea, and what we wanted to see is a difference between observational data and our offline funnel model. This was published, I think, in July in Ocean Modeling, in the Ocean Modeling Journal. So here we see the area average surface chlorophyll concentrations from the offline funnel model in blue, and in green we see satellite observations. And we divide the domain. So we had the full domain, and then we have the Catalan-Balearic Sea, which is, well, the Catalan-Balearic Sea. And then we have the southern part, so it would be south, the Balearic Islands. So we see that the model captures the seasonal variability well, but we have a small offset in the southern part of the domain, which is a bit strange. I won't go deeper into that, but we can see that the model with observation agrees well. We also did a comparison with biogeochemical Argo, and we see that in the different seasons, and we see that chlorophyll shows good correlations in winter and spring, especially in the Catalan-Balearic Sea, which is this A part, so the northern part of the domain. And nitrate performs better, especially in the southern subregion. So here we have these animations of monthly concentrations of ammonium and phosphate. And we see that the model is able to reproduce mesoscale structures, and this is consistent with the described regional dynamics in the literature. So this was just to include a visual example that the model can capture the physically plausible processes that occur here, like, for example, these mesoscale eddies. We can see also the northern current that comes from the Gulf of Lion and goes down the Catalan coast. Yeah. And then I wanted to show, this is not published, it's a work in progress. This is a res activity, long-term activity I had last year. So I wanted to test the climate projections because I needed to. And what I did is implement a configuration of 2-kilometer resolution. So again, in the northwestern Mediterranean Sea, you can see the domain here in the left. And what I did was use the atmospheric and riverine forcings from the CMIP6 experiment. It is from this, I think it's a French institution, I can't remember the name, Centre National Météo France. And what I did was I ran the model in the historical scenario from 2000, from the 2000 up until 2015. And then I run two future scenarios. So the shared socioeconomic pathways, I run the sustainability one and then the middle of the road until 2034. And here you have some example because I'm still processing and analyzing the data. It's taking me a lot of time, which I didn't expect that it was gonna be that hard. But I'm only me working with this, so it's a lot of work. So here you can see the spatial anomalies from, this is a snapshot because I still don't have all the data processed. So this is from December 2034 minus December 2001, just in case to give like a spin up time. And this is a surface layer, and it's the SSP1, so the sustainability scenario. So here we can see the difference in temperature. For example, we see in this specific snapshot, 4 degrees temperature increase from 2034. And we can see that there's a decrease in chlorophyll and phytoplankton, also in nitrate. We see it here in the Gulf of Lion region. So we still have to analyze all of these. But the difficult part, which are the simulations, is done. And yeah, I'm very looking forward to analyze all of this. So as a summary of the offline funnel model, we see that this decoupling, we gain around 80–90% of computational reduction. We maintain biogeochemical fidelity. We can see it with the Gulf of Mexico model evaluation. And this enables us to do long-term projections, which are faster. And then we can do this sensitivity test under climate scenarios, because we don't know if, well, I don't have to explain this to you, but yeah, we need to do a lot of tests. So this is very easy for modelers to implement, and then you don't lose a lot of time in the end. Does this make sense? So yeah, this is related to my objectives of assessing limitations and then develop these new approaches, analyzing, evaluating them, and also consider this climate change dimension problem. Then I will present the HypeCafe model, which is a hybrid model, biooptical, that I developed at the Max Planck Institute of Biogeochemistry in the Atmosphere, Biosphere, Coast, Climate, ABC3 Cube research group with Alex Winkler and Vitus Svensson. And this is still in preparation. But basically, this simple conceptual figure shows us that we have in situ data. So we want to use the in situ, the real measurements of marine primary production, not that we have derived products. So a direct measure. We feed it with remote sensing into a machine learning algorithm, and we merge it with the CAFE model, which is the absorption model that I presented in the first slides. It's the carbon absorption fluorescence zeophotic resolving model, which is considered to be one of the best ones that we have of the bio-optical algorithms. We merged it and we did the HypeCafe model. Very cool name. It means hybrid primary production estimates based on the CAFE model. Okay. So here we have a scheme of, I wanted to say something else, sorry. Well, it's okay. Here you can see that it's PyCafe and not CAFE. So the original CAFE model is in C. And we wanted to make this tool user-friendly, accessible, open, and yeah, also more compatible with data-driven methods. So we said, okay, let's make it Python. So now it's PyCafe. It's open and it's available if you want to have a look. So here we have the cafe model scheme, which includes environmental variables like PAR, latitude, mixed layer depth, these absorption properties like absorption from phytoplankton, chlorophyll, Gelb stuff, base absorption, sea surface temperature. There's all of these processes that I can explain later if you have any question. And then ultimately we have NPP, net marine prime production. So the final equation in the model would be NPP equals the absorbed photons by phytoplankton multiplied per the quantum maximum efficiency of phytoplankton and the hyperbolic tangent of spectrally resolved irradiance and irradiance divided by irradiance. Well, light things, okay? So we said, okay, we can simplify this and we say MPP, so marine prime production equals the absorbed photons and how phytoplankton uses these photons, this light. So we will call it light use efficiency. LUE. So it's the researcher that made the model in 2016. This July published a paper, and he also made this reduction, but they call it photosynthetic efficiency, phi mu. It's the same, but when we designed it last year, this terminology was still not there. So I call it light use efficiency, photosynthetic efficiency. I'm still trying to remember to call it photosynthetic efficiency so it's consistent with literature. But just in case I get confused and I say LUE, it's because we started saying LUE. So we say it's an absorption model. So we say the observed photons must be
Julia está silenciada.
Okay, now?
Ahora sí.
So here we have the scheme of the CAFE model. And summarizing, we have this equation which of the model, which is MPP equals the observed photons multiplied per how phytoplankton uses these photons, so how it uses this light. We simplified it and we call this parameter light use efficiency or photosynthetic efficiency. So we rely in the CAFE model and we keep the absorbed photons variable. And then what we learn is how phytoplankton uses this light. So this light use efficiency. To obtain this light use efficiency, we divided the observations from NPP, from the Mattei and Scardi dataset divided by the absorbed photons from Mattei and Scardi. But we had a problem. And is that most of the in situ measurements are from the pre-satellite era. And you know that for a machine learning or data-driven algorithm, we need a lot of data. So we designed this workflow, which is a bit complex, but I will try to be quick and concise. So what we did is to design another machine learning algorithm, which is a gap filling optical properties, we called it. It's a random forest, and what we did was to train this random forest with satellite data from 2003 to 2023 from environmental variables, so the target were the optical properties, and the input variables, it was PAR, mixed layer depth, chlorophyll, and sea surface temperature. Because we are lucky that the Mattei and Scardi dataset includes these environmental variables. So we trained these, and we got 0.99 R-square. So this gap filling optical properties algorithm was really good. So then we could run the CAFE model in the Mattei and Scardi locations. So this is another version of the model we did. And is that the model now can be run with NumPy arrays. So this is also very useful for people that it's not familiar with NetCDF or Dask arrays. This is not published yet, but it will be very soon. So we run the CAFE model in the Mattei and Scardi locations. We gap fill these optical properties, and then we can train on LUE, on light use efficiency. So we did 3 final global products in 9-kilometer resolution. We have the PyCafe outputs. Then we have our hybrid MPP. So with learned LUE and our absorbed photons. And then we said, okay, let's run a simple random forest. The input, the predictors are the input variables from the CAFE model, and the target is NPP. Maybe we don't need to do all of these things to have this improved final product. So we have these 3. How is the performance of models? So here we have scatter plot. In the x-axis, we have the target, so measured MPP, in situ MPP, and in the y-axis, we have the predicted MPP by the different models. In circles, we have the PyCafe, so the absorption model, which is considered one of the best. Then we have in triangles the random forest, and with orange stars, we have the hybrid. So as you can see, the PyCafe model is very far from in situ observations. Then the random forest is better, 0.79 R square, and the Hype Cafe is a bit better. But the thing is, Hype Cafe, it gives us some interpretability. So we chose to keep this hybrid version. Here you can see the spatial distribution, the global mean MPP from 2003 to 2023. And we have this in the three models. And as you can see, the PyCafe underestimates in relation to Hype and the random forest. Basically, we can see this in the equatorial band and also in the Arctic. I'm presenting some of the results. This is again another thing we did, is that we tried to do an emulation. And the good thing is that the cafe model is very good for emulation, which is very good for us because it enables us to do super fast runs. So to do tests and different things, it's very useful. We can see here that the R square, it's very good for both the validation and test. So finally, as a summary, we can see that this hybrid approach outperforms the state-of-the-art cafe model. And this feature importance analysis gives us new insights into MPP and also light use efficiency behavior. Why I'm saying this? We found that there's very different results in Lue when we compare the absorption, so the cafe model, and our data-driven model. We can see here that Lue in cafe, it's almost constant, but with low variability. And we see that there's an increase in light use efficiency in the hybrid model. So we are checking if Lue could be a potential ecological indicator of MPP in the ocean. Then we have some hypotheses. There's this paper from Davis et al. From May 2025, this year. They record that there has been a decrease in light. So the title of the paper is A Global Darkening of the Ocean. And then there's the stratification we all know about. So physiologically speaking, phytoplankton, when has less opportunities of light and nutrients, is more efficient. This is in lab experiments. We don't know, in like globally speaking, but we think that these two causes may be a reason of this light use efficiency increase. With an increased stratification, when phytoplankton has this opportunity of light and nutrients, it becomes more efficient. We are checking this hypothesis. But yeah, this relates to the objectives that I mentioned in the beginning. This is some work in progress that we are doing. So we took one step further, and we have done simulations with Hype, doing an ensemble of 10 simulations. And what we do is that we randomly add noise to the in situ observations, because we know that in situ also have some uncertainty. So this can give us an ensemble, so a mean result of hybrid MPP. And here I show some results of Lue and MPP in the different biomes. And we can see where these are more different, more similar, different trends, and so on. I think I don't have much time to explain this. This is in milligrams carbon meter square per day. Here I put some global numbers in case we want to discuss later. But we see that Lue might be an important factor. And now I want to explain the future work I will do very quick. which is the Fresh Care project that I will start in January at the Institute of Marine Sciences with the ERC starting grant from Marta Humbert. And basically this project is to implement, so my part of the project is to implement an sea ice coupled model to improve freshwater quantifications in the Arctic. So as you can see here, there's a lot of there's a lot of models that are already implemented in the Arctic, but when we see the observations, these are very far from reality. And when the quantification is good, then there's some issues, especially. So the Beaufort gyre is not well located, or we have the Beaufort gyre that it's circling the wrong way. So this is a concern, scientifically speaking, and we want to improve this. We want to improve this modeling part. So we are going to implement the ROMS model with the sea ice model from Dursky and Korokov with two horizontal grids with 25 kilometers and 5 kilometer grid, because there's some studies that show that with less, so with higher resolution, the results are worse. So we want to test. And the difference of our model and the ones that are already implemented is that this project, the Fresh Care project, will develop a product using deep learning of reconstructed in situ profiles. So we want to improve the boundary and initial conditions of this model, this sea ice model, and hopefully we will get good results of freshwater content and freshwater transport. And the outcomes are to publish this new high-resolution coupled model in the Arctic, and then validate freshwater content and freshwater transport in the historical period. And another thing that I forgot to say is that once we have all these configurations set up in the historical period, we will do like in the other work in progress that I presented, and we will run the model in the different SSP scenarios to see how it compares to the rest of the CMIP models. Hopefully, we will enhance the predictive capacity of Arctic freshwater dynamics under climate change. Finally, as a recap of marine primary production tools, we see that data scarcity remains a bottleneck for model validation, calibration, and training. I have been struggling a lot with this, so we still need more data. I think that synergies between these process-based models or biogeochemical models and machine learning models are key. We have been recollecting data, we have been improving models. Now we need a new thing to find novel relations, novel information that we still have not detected with conventional models. In terms of climate change, efficient frameworks like an offline or emulations are key in this context. We need fast and we need efficient and good tools. Finally, I wanted to say that both the offline funnel model is public, it's in Zenodo, and there's also an offline funnel guide in the papers that are published that explain how to configure your simulations, which is it's in an appendix, but I'm very proud of this because it's very difficult to set up a new model. So hopefully it will be useful for people that want to use this. And also the PyCafe model, it's available too with NetCDF or Dask. I still haven't published the one with arrays, but it will be maybe this week, I don't know. Depends on my work. And the Hype Cafe model hopefully will come very soon. And yeah, we are still analyzing data, so still a lot of work to do, but I wanted also to acknowledge the Agaur scholarship that helped to pay my contract, my PhD, and then the Institute of Marine Sciences, since I did some of the simulations, the first simulations of offline fennel in their cluster. Also, Red Española de Supercomputación and the Barcelona Supercomputing Center for hosting my climate projections. And finally, Fundació Universitària Agustí Pedro i Pons and Santander Bank for funding my research stays at Dalhousie University in Canada and in the Max Planck Institute. So that's it. Thank you very much. Hopefully, these models will be useful for the biogeochemical community. And I'm very excited to talk and to chat. If you have any question, please let me know.
Thank you.
Okay, brilliant. We'll move on to any questions here in the
Some
group.
question online.
I've already asked.
Okay,
If
I
there-
still
if there are, they were clapping online.
Okay.
They're still not asking